The Visual Language and Visual Learning Center is a National Science Foundation center. There's only 6 in the United States, and we are amazingly lucky to have one of them right here. BL2 has a variety of purposes. You know, for example, that most of the focus of research has been on the auditory modality, and by contrast, BL2 is focused on the visual modality. It's focused on what happens when the lion's share of our knowledge comes in through the eyes, from the lion's share of our social interactions comes in through the eyes. What is the impact of that on brain structures? And in turn, what is the impact of that on brain functions? And we're especially interested in the function of early language acquisition, of reading, and of literacy. The Brain and Language Laboratory at Gallaudet University is a very special lab. It is a laboratory dedicated to neuroimaging studies about the function of very specific brain tissue. So what's extraordinary about this particular laboratory, the Brain and Language Laboratory, is that it's a research-dedicated laboratory on a campus that has undergraduates and graduate students. Another rare factor of this laboratory is that young undergraduates can learn neuroimaging with among the best neuroimaging systems we have in the world. So I'm very excited about the BL2 laboratory because it makes available an extraordinary type of training to young people that's considered rare even in the United States at other universities. One of the things that's very important about what we study in the laboratory is we study the human development across the lifespan. So we look at very young children and how they process language, and then how they then later learn to read. We follow children through the reading acquisition process and look at what is most optimally beneficial for learning to read and the relationship between their reading and the human brain and what's best for both together. And we also look at adult processing of language. The other thing we do is we look at bilingualism, and we look at the impact of the human brain being confronted with 2 languages as opposed to 1 language. And that provides very important insights into the timing of exposure to 2 languages, when is best for a child to be exposed to 2 languages. So we've had several lines of research. A new line of research now is we're testing hypotheses about auditory tissue development. And in particular, we've looked at the question of whether early exposure to sign language interrupts in any way the typical healthy development of auditory tissue. And this is a very controversial topic. This is a topic that constitutes some of the common beliefs that people have out in the public, and we thought it would be very important to test this empirically. Up until now, there have been no direct tests of this hypothesis because there hasn't been the technology available to test this hypothesis. So there's this interesting relationship between the development and advance of a technology and the new types of questions that it permits us to answer. And so now, finally, with the advance of this new technology, we can look into the brain and evaluate these hypotheses in a more direct way for the first time. We've been very excited to find that early exposure to sign language is actually optimal, and that with early exposure to sign language, the child's brain develops in a healthy and robust way. Ironically, it's the children who were not exposed to had early exposure to sign language where we see aspects of deviation in the maturation of tissue that's participating in language. And we've been able to identify that for the first time using this brain technology. The system that we're using now is called Functional Near-Infrared Spectroscopy System, and it's among the most advanced neuroimaging technologies in the world.
Here we are in the BL2 with our very own FNIRS. FNIRS stands for Functional near-infrared spectroscopy. We call it FNIRS for short. The FNIRS is a special kind of technological device that helps us to observe activation in the brain. It does so by using light signals. You may wonder how light signals help us understand what is happening in the brain. Well, the use of any part of our bodies, such as our arms or legs, requires oxygen. That oxygen is carried to the specific area through our bloodstream, and the oxygen helps us use that part of the body. The same thing happens in the brain. When we use a specific part of our brain, oxygen will be carried to that area through the blood. Light signals are reflected more strongly when there is a high amount of oxygen present than when there is a low amount.
FNIRS is the only neuroimaging system that permits the study of both language perception and expression. For example, both hearing and producing spoken language, or seeing and producing sign language. We can accomplish this because the system can tolerate moderate movement, unlike other neuroimaging systems. And because this allows us to study both the perception and production of language, we are able to see a fuller picture of human language.
The FNIRS helps us to understand how the brain processes language, whether it is spoken or sign language. The fMRI only captures language perception, while the FNIRS is portable so it can be moved, and the sensors are lightweight so they can move along with the participant's head. Because the sensors on the participant's head allow for movement, moving right along with their body, we don't have to limit their movements. Also, the FNIRS helps us study how two people interact with one another at the same time.
FNIRS permits the study of the brains of very young infants, which is not typical of other neuroimaging technologies such as fMRI. Unlike other neuroimaging technologies that produce magnetic or electrical fields, which excludes the study of children with cochlear implants, FNIRS uses light, which permits the study of individuals with cochlear implants from very young ages through adulthood. Because of this new ability to study a variety of populations and people of many ages, the FNIRS system has revolutionized the study of human language acquisition in the growing brain.
Something extraordinary to note is that Gallaudet University is the only FNIRS center without a partnership with a medical center. Most universities with an FNIRS are required to collaborate with a medical center and share the equipment with medical personnel. At Gallaudet, we have research-dedicated neuroimaging, which is incredible.
Another thing that I like as a neuroscientist is that this particular system is a faster measure of neuronal activity than contemporary fMRIs. So the typical scanning machine takes snapshots of the human brain once every 2 seconds. Now consider what this system does. This system takes snapshots of the human brain's neuronal activity 10 times a second. So you can imagine that gives us a closer measure, a more accurate measure of neuronal activity than the fMRI. So we are very lucky to have this system. We are able to answer brand new questions about the nature of human language acquisition that we could never answer before because we weren't able to see into a child's brain under 5 years old before. So this allows us, for the first time, to study the development of the brain tissue over early life, the development of language over early life. For the first time in history, we have a window into the most early entry into the human language acquisition process. We're in an interesting point in our understanding of sign languages and an interesting point in history. I think it's fairly common that most people and public in the street, if you stop them, would say, oh yes, sign languages are real languages. We know from 40 years of linguistic research, very beautiful linguistic research, that sign languages are in fact real languages. And I think most people in the public understand that people who use sign language constitute a distinct culture, that at the social, cultural level, these are unique groups with integrity and a coherent culture. One of the things I find rather surprising as a neuroscientist is that people will hold those two views. Sign languages are real languages, and sign languages make up groups of people who are distinct cultures. And at the same time, you reach a wall. And the assumption is that, yes, sign languages may be that, real languages and constitute a culture, but it can't possibly be biologically equivalent. And I've always been fascinated by this notion that somehow sign languages are not biologically equivalent. And if you say that another way, the view is that human language obligatorily requires speech, and that somehow speech is special, and that speech is more special to the human brain. And so for many decades now, I've been intrigued by that hypothesis and set studies to test the hypothesis. And so one of the first assumptions about speech being special is that it's absolutely essential for human language acquisition, that the presence of speech is required for a child to reach normal maturational milestones. So in my laboratory, we tested that first assumption, a common belief that speech, the presence of speech, is absolutely essential for human language to proceed in a typical, everyday, normal way. And so we looked at groups of children exposed to sign languages from their deaf parents early in life, and groups of children exposed to spoken languages from their hearing parents in early life. And so if the assumption is correct, if speech is absolutely obligatory for achieving normal language acquisition, normal milestones, well, we should see the signing children fail to achieve the typical universal milestones that are known to exist in human language in the species, the early milestones that most parents could identify, the early babbling at around 10 months, the first word at around 12 months, the first two-word combinations at around 18 months, and then the rich embellishments that follow 22 months and 24 months and beyond. And so when we compared the two groups, signing children and hearing children, we found that all children, regardless of their language input and the language modality, all children reached every milestone on the identical timetable. We then looked at other children. We looked at groups of children who were coders, hearing children of deaf parents who were getting two languages, both a sign language and a spoken language, perhaps spoken language from a grandparent and signing from the parents. And there again, we tested the hypothesis that speech is special. Speech is biologically special. If that were the case, a young baby exposed to speech or sign might be expected to turn away from the sign input. If sound and speech were special, you might expect that child to glean any morsel of sound they could possibly get. And so you might expect an asynchrony, a timing difference in the child's achievement of the speech milestones, which should be normal, as compared with the sign milestones. Instead, what we found is those children achieve each and every milestone on the identical timetable. And then we tested that hypothesis one more time with a very extraordinary group of children. These were hearing children, typically hearing children. Nothing was wrong with their ability to hear sounds in spoken language. Yet they were being raised in homes that had rich language traditions, except that it was in sign language only. These children were receiving either American Sign Language from their profoundly deaf parents, or they were receiving another sign language that we studied, which is Langue des Signes Québécois, LSQ. And that's the sign language used in Quebec, in Canada. And so across two countries, these groups of children who were only being exposed to sign language, who happened to be typically hearing children, you might expect would have very deviant language acquisition if speech was biologically privileged or special. You might expect that child to have all kinds of deviance, social, cognitive, linguistic, but certainly the view that speech is special would not predict normal development. It turns out the children were entirely normal in their language acquisition, milestone for milestone. Developmentally normal too, cognitively, linguistically, socially normal. These children took the sign language, used it as a real language, the brain showed normal development, and so we have a very dramatic example where speech was ostensibly special, and yet the children showed us that that's not true. that it's language that's special, not the presence of speech, not the modality. In fact, what these studies, these three groups of studies taught us collectively is that the human brain is not looking for very specific modality like sound. Human brain is looking for the patterns that are pressed in human language, and we have actually privileged tissue in the human brain that's set up to find those patterns in the input. And it's the patterns that the brain wants, not language on the tongue or language on the hands. The human brain wants the patterns that are in natural language. So we thought, well, we can put that particular hypothesis to rest because the data didn't support that. The second hypothesis had to do with whether or not a common belief is that human speech is absolutely essential for the human brain to have typical brain organization, to have typical tissue development, to have typical processing of human language. That was thought to be absolutely essential. So the early behavioral studies that we saw in development didn't support that hypothesis. In fact, what it supported was a very radical hypothesis. The hypothesis it supported was that whatever is controlling the acquisition of speech is also controlling the acquisition of sign language. In fact, the brain must have a common set of tissue that wants a certain type of pattern. In fact, it's so powerful, this tissue, that it could perceive an invariance in pattern on the hands, an invariance of pattern on the tongue, and see through converging evidence that those patterns were the same and just learn language. So that led to the hypothesis that it's the same brain tissue that may be guiding speech and sign. Now, this was a very radical hypothesis at the time, because the tissue in the brain that's responsible for human spoken language is near the ear. People thought, well, of course, that has to be tissue that's unique to sound processing because of its location. It's situated near the ear. That tissue participates in the processing of spoken language. Therefore, the tissue must absolutely be unique to processing of sound. In fact, we went to try to stack the deck against us. We went to the unique parts of the brain that for 125 years, neuroanatomists have told us that tissue is unimodal sound processing tissue. That the brain, that's all that tissue does, it processes sound, and in particular, the sounds in human language. So we conducted among the first neuroimaging studies, brain scanning studies, and looked into the brains of signing people and of speaking people. We looked at people who were deaf and signed all their life and people who were hearing and spoke all their life. And we gave them parts of human language that were unique to very specific levels of language organization. So we gave them aspects of what's called phonology, the small set of meaningless units that we combine over and over and over again according to the rules of our language to make all the words and sentences of human language. And it's called the phonological level of language organization. And people said that's unique to sound, it's near the ears, it's nothing else that tissue can be. And so we gave deaf people the equivalent phonological level of sign language. These were bits and pieces of signs that we draw when we make up signs that we use to make the sentences that we make up in signed sentences, but then that in themselves have no meaning. They really are equivalent to the phonological level of human language. And what we found is that the identical brain tissue was governing that level of language organization that had universally been regarded as being unique to sound, and moreover that had universally been regarded as being processed by tissue in the brain that's only responsible for sound. And so these were quite extraordinary findings because of a lot of things. For one, the deaf people's brains were not processing sound. So neuroscientists had previously believed that tissue should have shriveled up and died. That tissue should have atrophied. That tissue should have, because it didn't get sound stimulation, should have just shriveled up and retreated and not been functioning. But instead, not only was the same sound processing areas alive and vibrantly well in a deaf person's brain, but it was doing the same functions for language. So the sound, the tissue that is traditionally associated with phonology was doing the same job in ASL and LSQ. The tissue that's responsible for semantic processing of meaning was also processing semantics in sign language. The tissue that's participating in the processing of sentences in human language also processed that by the same tissue in the signing deaf brain. So that taught us that the brain tissue that we previously thought was only special to sound was instead not processing sound, but once again from the brain now processing the patterns that are in sound or sign. That the reason the tissue did not shrivel up and die in the deaf brain is because it was never dedicated to sound in the first place. It was dedicated to the patterns that are in human language, all human language, be it language on the hands or language on the tongue. The brain itself was not looking for sound. The brain was looking for patterns that are in human language. Another common belief is that early exposure to two languages is somehow detrimental to healthy language growth. And I actually see this a lot in my own laboratory because we study the bilingual brain and we study how two languages impact brain development in relation to children's dual language skills and in relation to learning to read in two languages. So the common belief is that somehow early exposure to two languages causes children to be either language delayed or somehow fundamentally confused. And this is a very deep and pervasive common belief. It's something that actually permeates our school systems. In fact, the way in which language introduction is in typical public schools is actually built on this assumption. That early exposure will confuse a child. So expose a child later when it's safe. So when the assumption is when their first language is stabilized, and then that it can tolerate the kind of assault or the contamination of the introduction of a second language. What we have found, by contrast, is that that particular view flies in the face of human biology. The human brain wants language, dual language exposure early in life. Early in life means really early in the early years of life when the child's brain's neural pathways are becoming set and established. And so we have done a variety of studies on this topic, 3 decades of it, and what we found is that the most optimal human language acquisition occurs from birth to age 5. And that if a child has two languages exposed to them during that period, rather than being confused or delayed, the human brain takes the neural tissue that's been biologically endowed for processing language and grabs it and sets it up and begins to build these pathways in two discrete systems. So the child is not, rather than being confused by early exposure, the brain sets up stronger, distinct language representations than if they had received language in a sequential or serial way. For example, exposure to one language and then later exposure to another language. In that second scenario, that is when we see children have high intrusion errors. That is when we see children have a laborful, effortful process of acquiring the second language that was exposed, the one that was exposed later. So it's contrary to the common beliefs. The data, the research shows us that languages most optimally have to be introduced to the child early in life and in the same developmental period. That's absolutely essential. I couldn't emphasize this more. This is from multiple disciplines, from multiple types of studies. This is among the strongest pieces of converging evidence we have in science. It's totally uncontroversial, which is rare in science. We know that early exposure to languages, early exposure to language is the absolute paramount, most important thing we can do for normal, typical development in the human child. We know this. We have this thing we call language, and it turns out it's made up of a variety of parts. And one of the most fascinating discoveries that we've had in the last 50 years is that each part of this thing we call language has highly specific brain tissue to do the job. So there's very specific tissue that's dedicated to how we understand sentences, and there's very specific tissue dedicated to how we understand the meanings, the small meanings in words. And there's very specific parts dedicated to processing the little bite-sized nuggets from which the nuggets that we call phonology, both in sign language and spoken language, from which we build all the sentences that we'll make in our natural language. And each of those parts of the human brain that are dedicated to the language function are on a very specific maturational timetable. The brain wants the experience and the exposure to the natural language within a certain developmental window. And the deaf child who's had early exposure to sign language has the relevant grist over which that machine is working. It's getting the right parts and bits and pieces that can then set up the categories of language in their brain that tissue's in a ready state, ready to process. So early exposure to sign language is absolutely essential because the brain needs to experience those parts of language. And if you don't have that experience, then you will create deviance in that brain tissue that is sitting there waiting and ready to receive that information, but you didn't give it to the child. There's another version of the common belief about bilingualism, and that has specially to do with parents who have deaf children. And here, the way it gets played out is that there's a worry that children early exposure to sign language will somehow hurt the child's acquisition of English. At the core of this, you can see the connection with a worry about bilingualism. But now it has to do with, if you're exposed early to sign language, that it will interrupt and disrupt a child's acquisition of English. And so there's been a practice of withholding sign language from young deaf children. And we've actually tested that empirically in the Visual Language and Visual Learning Center in BL2. In fact, 15 labs across the United States have asked this question in one way or another. And so to give you the bottom line, spanning these labs through converging evidence and different technologies, we have found that children who are exposed to sign language early in life are actually stronger in their acquisition of language, stronger spoken language, and here I'm talking about English, stronger in their acquisition of reading English as compared to children who are deaf who've had only spoken language instruction. And so why might that be? And we can actually track over development exactly why this is so. So we have found that deaf children who are exposed to sign language early in life, in the experience with natural sign languages, very early in life begin to have changes in their visual systems that allow them to be better at tracking eye gaze and the relationship to where an adult is looking and the things that they're referring to in the world. So just think about this. If you're a baby and you have a stronger capacity to follow visually where your mom or your dad are looking, you are going to take in a stronger way, early vocabulary learning. Well, if you have a richer vocabulary, you will then evolve into having a stronger language utility, language ability. Following from having stronger vocabulary and language facility, these children, when they go to learn how to read and write in spoken English and knowledge of the spoken language, transfer the strength of that incredible foundation into cracking the code of English. And ironically, those children who have had early exposure to sign language are actually stronger readers in English than children who are deaf and who have not had access to sign language. And they have shown to be stronger readers as compared to monolingual hearing English children. This is a very powerful finding. So the children who have had early exposure to sign language develop linguistic and cognitive advantages that permit them to more powerfully crack into the code of learning spoken language than other groups. In a more contemporary way, there's been a new hypothesis that's been offered, and this is specifically about young children who are deaf, who have had cochlear implantation. Here, the hypothesis offered is that if a young child has cochlear implantation, that you should not expose the child to sign language, because somehow the belief is that this will hurt auditory tissue development. That somehow, if you have a CI, a cochlear implant, that the child should not have exposure to sign language because that will interrupt typical healthy development of the auditory tissue that will permit them to learn language, to learn spoken language, to learn English. We found that's false. One of the reasons why we weren't able to answer the question before now is that we didn't have the brain technology to look into the brain and track very specific tissue and follow it over time. Now, with our new technology, we are able to look into the brain and see, is that the case? And with the new technology that we have here at Gallaudet, we have studied people with cochlear implants who've had early exposure to sign language. And we've compared them to adults who've had cochlear implants, who've had late exposure to sign language. And what we have found is that people who have had early implantation and early sign exposure have entirely normal and typical auditory tissue development. That sign language exposure early in life with a cochlear implant did not hurt the development of auditory tissue, didn't interrupt it, didn't impede it, didn't cause it to be deviant in any way. By contrast, people who had cochlear implantation and did not have early sign language experience, early sign language exposure, they show non-standard organization of the auditory cortex, non-typical with regard to comparisons to a hearing person's use of auditory pathways. So ironically, the group who had early cochlear implants and had early exposure to sign language had typical auditory tissue development. Early sign language exposure did not hurt the development of the auditory tissue in any way. In fact, they were the group that had the most normal, typical auditory tissue maturation compared to all the groups. So why did these people have better auditory tissue use? The auditory tissue use that we're talking about is not dedicated to sound. We have made a mistake. We believed that the auditory tissue was only looking for sound. We now know the auditory tissue is looking for highly specific rhythmic undulating patterns that are pressed in sound and pressed in sign language. The tissue it's a misnomer. We're calling it auditory tissue. The tissue can process auditory tissue if it's given auditory stimulation. But the tissue can also process visual information if that's the modality within which the patterns are entering the brain. Because the reason that the group of people who had cochlear implantation and sign language exposure show normal auditory tissue development because they got normal language input. The patterns on the hands allowed the tissue to stay alive because the tissue is not based on sound, the tissue is based on patterns that are pressed in language. So in the auditory tissue, ironically, if you don't give it the right pattern, you could interrupt its normal and healthy processes. So now we look at cochlear implant for a moment. A cochlear implant is not buying your child a new ear. The child has to learn how to use the stimulation that is coming in from a cochlear implant. And that takes time. And so in that time, the child is not getting relevant patterned information.
The discovery of the biological equivalence of speech and sign has important and far-reaching implications. Most loving parents would never withhold food from their child. We understand that this has negative impacts on the child's biological development. Similarly, there are negative impacts on the biological development of the child's brain if early sign language exposure is withheld from young deaf children. And this includes young deaf children with cochlear implants or hearing aids who are receiving early intensive speech instruction. These biological impacts on early brain development in turn have implications for the brain's higher cognitive functions. For this reason, decades of developmental cognitive neuroscience research teach us that exposing a young deaf child to sign language in early life is a biological imperative on par with the biological imperative of providing our children with food.
Withholding sign language from a young child is withholding information that the brain needs at very specific times. You will impact the brain. You will impact the child's maturation of language because that is fixed and needed in human development. The human brain needs early exposure to multiple languages for the child to set up distinct and independent representations and for the child to achieve the most complete an optimal dual language mastery and success.
In summary, the brain is looking for patterns at the heart of human language, and it is looking for them early in life. Perhaps the most revolutionary implication of our work is that the brain does not discriminate between speech versus sign. It will take the hands if you give it sign language, it will take the tongue if you give it spoken language. Instead, we humans discriminate with our minds. It's a human tendency. But the good news is that the brain sciences have taught us that we have the remarkable human capacity to adapt and to change in the face of new knowledge.